And good morning. Yes, that's good. Now, I actually am replacing some lady professor from MIT, but I decided rather than asking someone else in such a short notice, I have to do it myself. Okay? So I didn't have very much time to prepare. However, the title of my talk today is Left and Right. Why does it matter? Okay? Scientists sometimes use very strange words. So we have to learn it very quickly, but it's not that difficult. Chirality. Chiral, achiral, enantiomers, diastereoisomers, racemic, racemate. Oh, very strange. But there are reasons. If you know the origin of these words, it's not that difficult. And you learn it very quickly, okay? What is chirality? This is a definition. An objective system is chiral if it is distinguishable from its mirror image, or if it cannot be superimposed onto it, or a mirror image of an achiral, that means non-chiral, object cannot be distinguished from the object. Now, I give you a very simple example. If it's a two-dimensional thing, scientist, okay? If you make a mirror image of this, it becomes like this. Yes. I looks ordinal, but all others are reversed. So it's distinguishable. So I is non-chiral, and others are chiral. However, we normally define things three-dimensional. We live in a three-dimensional world. So let's look at chiral thing. Hand, foot, and shoe. Yes, right hand becomes left hand in the mirror. So they are distinguishable. So they are chiral. Foot, shoe also. How about molecules? They are chiral molecules. If you look at this, this is amino acid. Ada and Penonera talked about this. L amino acid, D amino acid. Well, but that's D and L. Again, we use strange symbols to tell this is left or right. D, dextro, L, levo, means right and left in Greek. R and S, rectus and sinister, that is right and left in Latin. That's all. And achiral molecules and its mirror image are called enantiomer. Well, again, strange word, but actually this means in Greek opposite form. That is enantiomer. So it doesn't say really very strange thing. So this L amino acid and D amino acid are enantiomers, each other. A racemic mixture or racemate is one that has equal amounts of left and right-handed enantiomers. Because the work was done, actually Pasteur, long time ago, 1848 in fact, they used racemic acid because he worked on the fermentation of grapes. And it's called bunch of grapes in Greek, in Latin, it's called racemus. And racemic or racemate comes from that. And because they are achiral things, sock, animals' outlook, molecules, all these are achiral or non-chiral. So you learned all this. But what is chirality? It comes from this Greek word, chir, that means hand or foot. You know, if there's a chiromancy, look at your palm and tell your future. That's a chiromancy. You have some problem in your foot, you go to chiropodist. The same origin of words. Okay? Then, who defined it? In 1884, a man called William Thomson define that. The one definition I gave you in the first slides. Who is William Thomson? Do you know him? He is a physicist. But do you know? You know him by different name. Lord Kelvin. Huh? Do you know Kelvin? He is the absolute temperature's unit, Kelvin. K, that's him. When he was knighted, he got knighthood, instead of calling I'm Lord Thomson, he said, I'm Lord Kelvin. There may be J.J. Thomson or there's some other famous Thomsons. I don't know. I really want to ask him, but a bit too late to ask. But he's Lord Kelvin. Where this Kelvin comes from? You know, you always question, because I'm full of curiosity. Why? Now, this is the Glasgow University. William Thomson, equals to Lord Kelvin, was a professor of physics at Glasgow University, that's in Scotland. Yeah? And there's a river, stream called Kelvin, flowing inside the campus. You know, it's bigger campus than here. So there's a river called Kelvin. And he took this river's name, Lord Kelvin. So he fancied some other name. Our unit of temperature may not be K. Okay? So you learned something. Now, I'm interested in all these left and right. Symmetry, asymmetry. This is in Notre Dame in Paris. Oh yes, this is Uji Byodo in Hodo. This is in Kyoto. Look at this. You have a mirror image in the middle, but also it's reflected on the pond. So it's another mirror image. Isn't it nice? But if you go to Ginza in Tokyo, you see this sort of building. Wow, this is chirality. Yeah, so you have to go and see. And this is a small packet of sweets. This is also chiral. Whenever I see it, I cannot help by buying these things, you know. Not because I'm interested in these sweets, but I'm interested in these packets. It's really interesting, isn't it? This is chiral. Now, if you want to go up a little higher, you may use ladder, or you may use staircase. This is the spiral staircase. There is no chirality. Ladder is achiral. However, this is the photograph I took this July in Manchester. There is a National Graphene Institute. Professor Grime got Nobel Prize in Physics on his work on graphene, and they celebrated his Nobel Prize in Physics and made this institute. And I had a chance to visit it this July in Manchester. Just mirror imaged it. So staircase or helicity, it can be left-handed or it can be right-handed. Yes. But I'm interested in chirality in nature, actually. My research is chirality in natural things. And this I drew. This is the size, order of magnitude. This is a bigger one. And in logarithmic scale, 10 to nth meter. 2 is 100 meters. It's like a whale. Okay? We are about 1.5 to less than 2 meters. Being not installed, you may be 2 meters. But still, you're very close to 0 in this scale. If you go down to 10 to minus 10, that is angstrom. So the molecular level. So this is 10 to minus 15, you go down to elemental particle. Okay? On the left-hand side, sort of biological world. You had animals, they are bilateral, and always exceptionally exciting. I'm working non-bilateral one, this snail. There's left-handed one and right-handed one. But although we are bilateral, yeah, but my body, internal organ inside is chiral. I have heart on the left hand side. My gut coiled in one direction. Yes, yours much the same. Okay? And these are determined by gene. This gene called, because it's genetically determined, nodal or PTX genes, which may come up later. And also, these genes or proteins are polymers made up of very small unit. And that unit, actually, it contains some sugars and also amino acid you heard several times already this morning. On the right-hand side is a material world. Again, there's a chiral molecules and many, many molecules, billions of molecules, recognize your neighbors and make crystals. Okay? So I really want to find out the process, how they recognize, discriminate the handiness we call, the chirality of your neighbors, and how just the molecule that is a gene decides which chirality this snail can take. So that's something I'm doing. So living organisms, the molecules, protein, genes, embryos, molecules to crystals that I want to work. But I have not told you yet, left and right, why does it matter? I haven't still told you about it yet. Okay, so now I'm going to tell you why does it matter. Because biological world is homochiral. Homo means the same. Okay? Opposite word is hetero. So homo means the same chirality at the molecular levels. So at the molecular level, biological world is homochiral. What does it mean? This is DNA. I showed you ladder and staircase. If it's written like this is just a cartoon. So you heard from others talk, A and T, C and G, they make pairs, and all these three, we are recording one amino acid, and CGTA, all these four letters is important, and that carries genetic information. But actually, it's not this form that DNA exists in bodies, in human, not necessarily human, in all organisms. It takes this right-handed double helix. Why it's right-handed double helix? Why it's not left-handed like I flipped over this staircase? Okay, helical staircase? Because there's a reason. You won't see it from here, so I have to show you another picture of DNA. Now, here, in circle, there is a sugar called ribose. If it's a DNA, it's deoxyribose. And if it's RNA, it's called ribose. And as I told you, there's D and L. That means in Greek, right and left. They are mirror images of each other, but all organisms on the earth uses deoxyribose. That's the reason it has to twist right-handed direction. Okay? How about proteins? Yes, I told you already, proteins made up of 20 amino acids. Other also told you about it. And there's a D and L forms, but all organisms on the earth, including bacteria, plants, or animals, use only L amino acids. Okay? There's no exception. So, that is what it means. Biological world is homochiral. DNA made up of D-deoxyribose and proteins, L amino acid, and this is the reason. Okay? So that becomes important to think about origin of life, evolution of life, food additives, pharmaceuticals and agricultural chemicals, and so on. Now, so on the primordial Earth, why there are left and right molecules maybe equally, how it's evolved, chemical evolution or biological evolution ends up only L amino acid word or only D, deoxyribose, D-ribose nucleic acid word. We don't know. There are several interesting hypotheses, but no one can prove it. So it's really exciting, but I'm not going into details. How it's important for food additives, pharmaceuticals, and agricultural chemicals. To understand it easily, I made principle called shoes and socks principle. You never heard of it because I made it. Okay? Principle 1, if you put your left foot into a right shoe or right foot into a left shoe, you realize it immediately. No difference in the case of socks. Principle 2, if a toddler wears father's shoe for fun, for example, he or she cannot feel the difference because it's far too big. Number 3, a sock takes the shape of either left or right foot when it's worn, but if you take it off, it goes back to non-chiral shape. However, if you don't clean it, if you put it a long time, there's a hole in a big toe part, then there's a chirality you can induce. Then, this is the same thing in chemistry. Chiral compounds display chirality only when they interact with chiral compounds or chiral electromagnetic wave. Actually, this is what is diastereomeric interaction. If they interact with non-chiral objects, there is no chiral discrimination. 2, if order of magnitude is different, there is no chiral discrimination, even chiral objects interact. And 3, dynamic chirality can be introduced to non-chiral objects. You know, if you put the socks takes the shape of your foot, so it becomes chiral, but if you take it off, it goes back to non-chiral form. So that is, but dynamic chirality can be converted to permanent chirality when you make a hole in a big toe part, okay? Now I have to go quickly. So this operates in the world. So biological world is homochiral. That means shoe and sock principle 1 and 2 at least operate. So aspartame, if it's the L-aspartic acid and L-phenylalanine, 160 times sweeter than sugar, but if it slips to D, it's bitter. Or carbons, if one is caraway seed fragrance, one is spearmint one, just a mirror image of each other, because we are chiral, okay? All our parts at the molecular level is made up of chiral things. And the thalidomide is a very famous case. And one is sedative, this is sedative, but one teratogenic, and R doesn't seem to be no side effect. So because of this, FDA in 1992 made a new regulation to making pharmaceutical compounds. So this is Professor Nori, unfortunately he's not here, and his wife, and when he got the Nobel Prize. It is just like this. So that's important to make only one of the enantiomers by chemistry, chemical reactions. It is so important. But my research, I don't have very much time to talk about my research, so you have to invite me back again, perhaps. But I'm interested in chiral morphology. It's the word I coined myself from chiral and morphology. And this is the sort of logo mark. So you have this sp3 hybridization. This is the molecules, simplest molecules, and also snail, because I'm working both. So just skip this. So I'm working on this. I explained to you earlier on. So there's the two. I'm a chemist by training. So I'm looking at the chemistry, chiral recognition, generation, enhancement, and measurement in a solid state. And also, I work on organismal chiral morphology group. That is molecular origin of the chirality of snails. I want to determine the gene. So first chemistry. Now I'm talking very fast, but I have to talk even faster from now on. I'm working on solid state. Why in solid state? Because in the solid state, molecules are densely packed and their orientations are fixed. Therefore, chiral discrimination, recognition, generation, and transfer occurs very strongly in the solid state or only in the solid state. So I'm not doing like Professor Noyori synthetic work, what we call asymmetric synthesis. I'm not doing this. I try to separate enantiomers from the racemic mixture, one-to-one equal mixture, or develop unique solid-state stereochemistry, or develop spectroscopic methods to obtain chirality information in condensed phase, or use chirality information obtained by this, try to use it to reveal the structures. For example, I'm working on beta-amyloids, or for a little bit on alpha-synuclein, Penira talked about, and also and they try to link between macroscopic and microscopic phenomena. So I don't have very much time, just go quickly. Perhaps I talk only one or two to make crystals from solution. The famous one is Louis Pasteur. He found sediment of this contains some chiral compounds, and he actually got this. Pasteur's and Scotty got sock-type crystal, but Pasteur got shoe-type crystal. And it was found in the bottom of the fermentation vessel. And this is what happens. Over 27 degrees, you get a racemic crystal, and below that. And in those days, Scacchi worked in Italy. There's no air condition. Therefore, he couldn't approach this, what we call spontaneous optical resolution. And I cannot tell you in detail, but actually, I determined the crystal structure much later. These Pasteur's and Scacchi's crystal, and I found some quite interesting phenomena, but it's too advanced, I'm not going to tell you about it. Now, then, this secondary alkyl alcohol, that is known to be very, very difficult to separate. Even you work hard, you may get 48% versus 52% for left and right. However, if I do it, I'm lazy, so I'm not doing anything to the solvent, to the alcohol, but use the alcohol just as a solvent and dissolve simple amine and acid, and just you get crystals after 2 days, and you get 95.5 versus 4.5. So you can get incredible enantiomeric excess. So you don't do anything but just using this. So the way you think something, you get things so easily. Okay, and there is a reason. There's the helical point, but I'm not going to detail. And there is some interesting things. If you grind crystals, you get the color, yellow and white color, crystal changes to red. And if you look at it, from solution, you get red crystal. By grinding, also you get red crystal, but they are different. What difference is this benzoquinone is sandwiched by R and R, but here R and S. Why is the difference when you get crystals from just solid grinding from solution? Because there is a memory of the previous original structure. If you dissolve it, everything goes to nothing surrounded by solvent molecules, but in the solid state, you still have a memory. That's the important part. No, I had to go quickly, and it's not only organic compounds. You can do a similar thing to different compounds from metal complexes, and if you grind it, and you get this from solution, if you heat it up, you get one-dimensional, three-dimensional polymer, but if you don't do it from solution and you grind it from the beginning, you get the polymer. But if you leave this polymer, it absorbs moisture from air. It goes back to mononuclear complex. But if you heat it up, it goes to polymer. You can do it nearly 30 times I tried. And there are many interesting things like that. And also, someone said it's nice to have a cover of journals. So yes, I have to show off cover of journals. Now, I'm not going to talk about. It's batteries. It's not something. It's batteries may be out. I'm not going to talk about stereochemistry, but just give you quickly about the development of chiroptical spectrophotometers, which can measure solid-state samples, because you cannot measure solid-state CD by commercially available instrument. What do? You make instrument yourself. You have to make tools yourself. And so I made 2 and had a patent on this. And if you made this, you apply this to samples. That's how you start working beta-amyloids and others. But I'm not going into details because I tell you about snails as well. So there's various ways, and this is the beta-amyloid, Alzheimer's disease one, and this is a precursor. And if you measure with our instrument in 140, 142, if it's cut 2 amino acids down, it is prone to Alzheimer's disease. It's a familiar Alzheimer's disease. It says it's a family who has more Alzheimer's patient has this type. And you can look at it, 1734, and it will has already more beta sheet, even in solution. And from solution to film, solid state, it changes. Alpha helix is going down, the beta sheet increases. This is how it starts to plaques. Okay? Now I have to go. And also these are hornet silk, and you can see even in the solid state, there's alpha coil to beta sheet changes occurs. And also we just managed to obtain the CD of gels, and we can obtain something quite interesting, but it is not published yet. So, and also I had a horizontal sample stage, and then you can work a little bit like dynamics of protein folding using our horizontal sample stage instrument. Now, let's go on to the biology side for 4 minutes. Molecular origin of chirality of snails. I want to understand why the single gene determines the chirality of snails. Okay? Oh, this is. And why I worked on Rimoniya stagnasis, a very advantages. There's no, it's a unisexual, or its chirality determines very early, and also it says in 1920 to 26 to 30, it's by single gene, but this gene is Mendel's single gene, okay, because it's before DNA structures determined, but we could prove it is really single gene locus. Then we did, and we have to start from scratch, because I'm a chemist. I was a chemist, okay? To do this, I have to really struggle to how to breed snails and all this. I had to struggle. But then this first one results is dominant dexter and the recessive sinister embryos are not mirror images of each other. What I mean is, in all textbooks all over the world, these figures are shown. Dexter one, sinister one, 1 cell, 2 cell, 4 cell, 8 cell, these stages, they are mirror images. Another textbook said that. But I realized it is not true. You can see it, the top, it goes, this is the left-handed one, the bottom, the right-handed one. If it's from 4 cell to 8 cell stage, it becomes like this. And but if you look at carefully, there is no mirror symmetry. Mirror symmetry is broken here, and this is the very important step. So what you call spiral deformation and the spindle inclination only in the dominant dexter snail, and there isn't any sinister recessive one. And we could, for example, change this dexter one, very similar to sinister one, by inhibiting the polymerization actin. Okay? So we know actin, the cytoskeletal dynamics is important for the Huntington's determination. And I can skip this one. And so it's not mirror image at all, but it is mirror image if it's the sinister on the Pfizer acuta. This is another species. So it came out in the front page of current biology, and also Science wrote about it. So I'm quite happy, and it says even elegant new study. So I'm very, very, very pleased about it. Now, let's go. Then proofs the snail hunting is determined by a single gene locus. We had this back crossing. So we had to mate left-hand, right-handed snail. That is very difficult. But we could manage it, and I can tell you just the results. However, it took years to make this. We made congenic one, which contains 0.1% dexter genome, 99.9% sinister genome. If you inherit the handedness determining gene, that's the right-handed determining gene, within 0.1%, it produces right-handed snail. And it always does SDE, spiral deformation, spindle inclination. And if it doesn't, there's no SDSI, and it produces left-handed one. So I thought, if that rotation at the 4 to 8 cell stage, because the third cleavage stage is so important, wow, if we just push it gently. So we did it. And it's a very mad idea. But anyway, I did it. Then, so we're doing it this way. Okay, so this is a dexter one. We have to push it a bit earlier because there is SD and SI. And with the sinister one, we just start it from here. And we culture it. Now it's very difficult because you're taking the things out and manipulating how to get it back. That is very hard. But anyway, we did it. We managed to do it. I'll show you it. It becomes completely mirror image creatures. Okay? Amazing. It's very healthy as well. So normally, this produces sinister juvenile, sinister adult snail, sinister offspring. But if I manipulate it here, it produces dextralized juvenile snail and becomes the dextralized sinister adult. But I haven't done anything to the gene, anything to DNA. It goes back to sinister one. That chirality dictated by the gene. And the reason why it becomes is there is a nodal and the PTX and all these genes which determine our heart position or gut coiling. The same gene operate downstream in snails. Therefore, if you manipulate it, it should be like this. It goes to complete mirror image side. That's the reason we get healthy mirror image creatures. So, and I'm not going to in detail because time is going to run out. But I managed to identify the handedness determining gene people looked for about 1 century. And this is just the first page of paper. Now you can see it, published October 6, only 4 days ago. Okay, so before I couldn't talk about it. But also it's accepted in September. But I know which genes and what it's doing. So I have to tell you about it. And there are tandemly repeated genes next to each other. And for the sinister one, DR1, there's a frame shift mutation. So it's terminate. So it's end up very short protein. And for the DR2, they are tandemly repeated. Dexter and sinister, they have the same gene, same amino acid. But this is sort of stopped here. And this makes sinister stagnized sinister. It's really exciting. And there are many other things, but I think the time is going to up. So this is just appeared. So if I have a time with a Western blotting, this is the wish. But there's a paper which tells you completely wrong, bad mistake, I think. This is our result. Some produced this result and showing this is the reason of asymmetry, but that is not true. So you cannot believe everything which is written in textbooks, written in journals. You have to think yourself hard, okay? So this is more interesting, but what I want to say is nodal gene that starts at 7.5 days in the case of mouse. Many genes are operating, but for this snail, before your zygotic gene starts to be expressed, chirality is determined already. Okay, so that's the reason I fell in love with this. So this I've been working, and I have many, not that many perhaps. This is not really working, but for the chemistry group, I had the major workers, and there are people here. And for biology group, yes, they're the PhD student, master degree student, the technicians. However, this is the work. And also, Ada showed nice birthday cake, so I thought I have to show birthday cake. So this is the birthday cake my students gave me. And how many candles? I don't know. I don't want to tell you. So, thank you very much for your kind attention.
Thank you for such a wonderful and up-to-date discovery report. And any questions from students? Quickly lining up. Go ahead.
So, hello. Thank you for your great lecture. I was fascinated with your talk about chirality or Schumann-Salk theorem. So, I have a question about chemistry part. And how can I know the molecule that a molecule is left or right? So, like, if I don't know ribosomes first, and if I made a first contact with ribose, how can I say that the ribose is left one or right one?
Okay, ribose is a very huge molecule, so we know it's made up of L-amino acid. But if I go back to the first question, if a molecule is right or left, it's very difficult to decide. We know they are opposite. How to define which is right, how to define which is left. Sometimes it's a sort of semantic problems, but in the past, we decided, we made a rule, international rule. If you look at stereochemistry and from one atom to the central carbon atom, for example, and if you make three substituent, which is sort of made a bond to the central carbon atom, if it is heavier one, like if you have a, for example, cerium or then oxygen or carbon, you just make 1, 2, 3. And if it goes right-handed way, right-hand circle, we call it R. If it is the other way, we call it left. So this is the definition, but we have to define it internationally. So there are all these international ones. Based on the stereochemistry, it's very easy to understand. So R and S, that is a definition, yes. Okay?
But the question was, how do you know which is which?
Which is which? Well, you can determine by CD, but again, circular diachrysm spectroscopy, you can tell which is which. But the definition is different. Yeah. Okay, it's a very good question.
Hello.
Hi.
Thank you for your informative speech. And my question is, so there are millions of creatures in the world. And how can we prove that all the creatures have D helix in their gene? In other words, why? So isn't there any possibility for creatures with L type?
Okay, it's a very interesting question because I cannot test anything. What do you eat?
Rice. Eat.
Yes, rice or meat.
Yeah,
What
meat,
are they?
Yeah. So.
They are other living things. Okay?
Yeah.
If they are made up of mirror image, you
Oh.
cannot digest it. Okay? So if all creatures survive and they take up things as a nutrients and they maybe metabolize it, whichever we are or all these bacteria, it has to be the same chirality. Okay, one day I want to write an SF story. I'm some alien and try to destroy all the people on Earth. So send some creatures, looks like plants or animals we want to eat, but it's made up of D, amino acid. But it doesn't look all different, but if you want to eat it, we cannot digest it because our proteins to digest, that is called enzymes, that's made up of L-amino acid, and it can only work for L-amino acid. So perhaps, I don't think there is any exception if they are alive as organisms.
Very
Okay?
interesting. Thank you.
Thank you.
If most subjects or matters in the world are viewed as chiral subjects, such as chairs and like what we view, does that mean the most basic basic components, such as atoms, are also chiral?
That is a very good question. One atom is chiral. Yes, people say it. There's a matter and antimatter, they are actually real mirror image to each other. However, I'm not discussing about that level. In that level, one atom, we tend to say, if it's a spherical one, there's no chirality. But if you go down to elemental particles, elemental physics, looking at charm or quark and all these, then spinning directions different for one atom, okay? But it doesn't influence what I told you. So we are really discussing about the level of molecules. Molecules are made up of spherical atoms. When they make bonds, it becomes, the orbital is not spherical anymore, but that level we're discussing about. But it is a very good question.
Thank you.
I appreciate your informative lecture. I think you have done many experiments before. Then what was the most hardest data to take, or where was the hard process of experiments and inspection?
Okay, every experiment is hard. I'm telling you only the succeeded one. Behind that, there are many, many failures, okay? And as if I'm telling you, I'm doing this and it worked, I'm doing this, it worked. No, 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 no. It failed and failed, and you have to think hard. But if you want to find something, you're curious, you really want to do something, well, I may learn from my failure, and I will do it even better. So that's the way you can improve. Of course, there's always the case you cannot control things. And some obstacles, someone told me I had many obstacles because I was a female and not like these days. Even my ex-supervisor told me the best thing for women is to get married. And this is your PhD supervisor tell you. So it's really shock. But then I went to England. I couldn't speak English at all at your age. But because I went to England after I got PhD, then that broadened my horizon. So any small obstacles, good chance. Okay?
Thank you for your answer.
Okay.
Hi, thank you for your great lecture. I'm glad to hearing. And I've heard about thalidomide, which famous accident from my chemistry teacher.
Uh-huh.
And when I read your paper, got a question, so I would like to ask. Is it possible to use the property of chirality to change heredity? And is chirality related to twins?
It's quite a different level of twins and the thalidomide level. Thalidomide level, it's metabolites in our body, and that metabolites work against what? Not many people know about it yet. So that is one stage. Because we are homochiral being, so left and right molecules sometimes produce different abnormal effect. If you have a shoe, if your heel part interacts, it doesn't get very much different from left and right. But if the toe part interacts with the foot shoe, then there may be a difference, something like that. No, not all drugs, chirality is important. Well, it may not be, but you have to be careful. That is the 1992 FDA's regulation. About your twins, this is quite interesting because what's happened to the twins, not much known, but when I gave a talk in Paris, when I was given the L'Oréal UNESCO Women in Science Award, the president of French Academy of Science came up to me, how exciting your talk is. My grandson, twins, and one is situs invertus. Situs invertus means completely mirror image. Body, you had a heart on the right hand side, gut cord interaction. If it is completely mirror image, it survives. If only part of organs is mirror image, it dies, okay, at earlier age or in fetus. So he said, so your things, mechanical manipulation, does it explain? And I had to say, all these molluscans, spiralium, it does the spiral cleavage, but human beings or the Homo sapiens or even the other higher or the animals, they don't do this. However, the nodal and PTX, that part is the same mechanism operates. So it's not exactly related, okay? But it's very interesting. So
Thank you.
keep thinking about it.
Thank you very much.
Thank you for nice lecture. What attracted you to this field of study?
You mean chemistry or biology? Both?
Both.
Both, okay. I started as a chemist. This may be by chance, okay? Because I was interested in looking at the molecular structures. That means I want to do X-ray crystallography, which really the very basic important technique to tell you which atom is bonded to make a molecule and how it's packed. I was in Tokyo University, graduate school, and one research laboratory was famous for determining what we call absolute configuration. That is this really right-handed and this is left-handed by determining the stereochemistry of molecule. So they are actually working on left and right-handedness. So I went in their laboratory. Then my interest in chirality, left and right-handed develops. But then I went to England. Then I thought, oh, it's interesting. Oh, biology is homochiral. It's even more exciting. So your interest and curiosity develops into different research fields. Okay, so that's how I come across. But maybe if it's simple, answer is by chance. Okay?
Thank you.
Thank you very much,
Okay.
Professor Kuroda.